Hi, hopefully you're here to learn about Gencove's cloud software for sequencing data analysis. Today, we'll be focusing on analyzing low-pass sequencing data. As you know, we've internally developed these powerful and scalable tools to handle low-pass sequencing data. We've also developed infrastructure to enable our customers to easily use these tools by our software as a service offering. We've gone a long way to make it easy to use by providing a web dashboard and command line interface and enabling customers to directly integrate systems using our event-driven API. In general, you will be doing two main categories of operations within the system. The first one is getting data onto Gencove systems, and the second one is analyzing data and viewing the results. This is a great segue for logging into the system at web.gencove.com because these categories are reflected in the left-hand navigation. My projects will be the area where you can list and create your projects, which reflect the data analysis category of operations. As you can see, in preparation for this demo, I've already created a project and specified its configuration as human low pass. Once the configuration is set, it cannot be changed in order to maintain consistency among samples within a project. If you'd like to try out other configurations, simply create new projects. On the other hand, my fast queues is the area where files that you have uploaded to Gencove show up. We always recommend uploading with the Gencove CLI. It is a straightforward installation on any system that supports Python, be it Mac, Windows, or Linux. Simply enter pip install Gencove and that should do it. Once you have the CLI, you can upload FASTQ files from your local machine or massively parallelize uploads from a cluster of instances in the cloud. As example data, I prepared a pair of FASTQ files that I'll upload from my computer, as you can see here. As you can also see, the files follow the loose Illumina file naming convention for FASTQ files, where the file name starts with the sample identifier, followed by an underscore, and ends in underscore r1.fastq.gz or underscore r2.fastq.gz. You can also notice that we have one pair of FASTQ files per sample. If you have reads spread across multiple files, from multiple sequencing lanes, for example, they need to be merged into one R1 file and one R2 file. Luckily, gzip files can be easily merged without decompressing, so it's simply a matter of concatenating the compressed file. Following these simple conventions makes data ingestion very easy, and we highly recommend maintaining a consistent file naming scheme. Now that we've covered the basics, let's upload some files. It's as easy as running Gencove upload and giving it a location. After that, of course, we have to authenticate. The Gencove command line interface will recursively search the location that you provided and upload all FASTQ files that it finds in that directory hierarchy. Customers have been quite happy with upload and download transfer rates, since transfers go directly to and from AWS without making a detour to Gencove servers. Once these uploads complete, they pop up in My FASTQs. There, you can see that the sample identifier and R1 and R2 designations are correctly identified. Since this list can be quite large, it can be sorted, filtered, and searched as needed. Running analysis is as easy as assigning the upload to a project. Select the upload, click Assign to Project, select the project, assign it, review, and that's it. This can also be done automatically by the command line interface by providing the Run Project ID flag, which will automatically run analysis on all uploaded files by assigning them to the specified project ID. By assigning an upload to a project, it has been converted into a sample that belongs to the project we selected. The project overview page provides basic information about the project, like its name, ID, breakdown of samples that are running, succeeded, and failed, and a histogram of sequencing coverage for all samples. Below all of that, there is a list of samples that can be sorted, filtered, and searched as needed. And navigating to a sample that I have previously analyzed, we can see some basic information about that sample. First of all, you can see the identifier that we call the client ID. This is an identifier that is populated by the customer and can be almost anything. We don't impose many restrictions on these IDs. To the right of this, you can see the Gencove ID, which is an auto-generated identifier provided by Gencove and guaranteed to be unique across all Gencove systems, projects, and samples. For savvy users, this is a version 4 UUID. We can also see a history of the sample, and further down the page, we can see some basic quality control metrics. The first one of which is FASTQ file format validation, and then a minimum number of bases sequenced, a maximum number of bases sequenced, the number of bases that have been deduplicated and aligned to the target genome, which in this case is human, an estimate of contamination by DNA from other samples of the same species, and an estimate of sequencing read uniformity across the target genome, which is measured by known sites in a reference panel being covered by at least one sequencing read. To the right of this, you can see an ancestry estimate for the sample, which can be an important covariate in downstream analyses. And below ancestry, you can see calculated polygenic risk scores for select association studies, along with the percentile the individual scores belong to. Finally, at the bottom of the page is a list of file deliverables. Most importantly, the imputed VCF file and associated index files. But we also provide the input FASTQ file, aligned BAM files, ancestry in JSON format, polygenic risk scores in JSON format, CNV analysis, and a classification of sequencing reads that don't map to the target genome, classified by a tool called Kraken. All of this data can also be downloaded in bulk using the Gencove CLI by simply running Gencove download together with the destination directory and a project ID. I'll create a temporary directory here and simply enter Gencove download, the destination directory, and provide a project ID. We authenticate, and the download starts. Of course, these downloads can be sliced and diced by sample ID and file type. So you can select to download only a subset of the sample IDs and or a subset of file types, which in addition to providing filtering and decreasing download sizes, also allows you to massively parallelize them. As mentioned previously, customers have been quite happy with upload and download transfer rates since transfers go directly to and from AWS without making a detour to Gencove servers. Everything you've seen in this demo, and more, is also available via the Gencove API, since the dashboard and CLI directly use the API. All of this brings us to the documentation. It can be accessed using the Help menu in the dashboard. In the documentation, you can find useful information about Gencove software, but also links to interactive Swagger API documentation and command line interface documentation. Finally, if you get stuck, you can always reach out to us at support@gencove.com. This brings us to the end of the demo, and hopefully you've learned enough in order to use the software. So until the next video, happy sequencing!